Okay, so now we're going to talk a little bit about mutations, or I'm going to talk a little bit about mutations. Okay, so as you probably know, mutations are changes in your DNA sequence. So mutations are a base pair change or a base pair insertion, a base pair deletion. We're going to talk about all of those types of mutations that changes the DNA sequence such that the allele is now a mutant allele and it's not functioning properly a lot of times. So sometimes you can have a mutation with no phenotype, sometimes you can have a mutation with very damaging phenotypes. So some examples of human disorders that are caused by mutations are, for example, cystic fibrosis. This is a mutation in a structural protein. So the CFTR protein is a chloride channel in the cell membrane. And when this CFTR chloride channel is defective, then the mucus that is secreted has a very high viscosity. And this accumulates in the lungs and leads to respiratory infections. Affected individuals have very abnormal salt transport across their membranes. There is a high frequency of this particular disorder in Caucasians, and about 1 in 25 are carriers. So this would be something that you might potentially get tested for if you were to decide to have children, right? So you could go to the doctor and be tested. A lot of the commercial direct-to-consumer genetic tests also test for this to determine if you're a carrier for cystic fibrosis. So again, this is a DNA change in a gene that encodes, right, RNA that will be made into a protein that makes a chloride channel, right? So this defective chloride channel causes these problems. Another example of a mutation in a gene that causes a disorder is sickle cell anemia. So this is a defective hemoglobin molecule. So as you probably know, that these hemoglobin molecules are important for binding oxygen and getting that to the surrounding tissues and organs and things like that. And this defective hemoglobin cannot bind oxygen as well. And this is just a single amino acid substitution. So when we talk in just a minute about the different types of mutations that can occur, one type of mutation that can occur is just it changes the amino acid, right? So in this case, it just changes a glutamate to a valine. It changes the codon from a GAG in the mRNA to a GUG, right? Just that one change causes this major issue in the binding of oxygen, and it causes these cells to be sickle. These sickle cells don't live as long as normal, and this seems to be in high frequency of those of African descent. So those are just some examples of mutations and how the changes in the DNA can lead to phenotypes that we see, disorders that we see. And so what we're going to talk about in this set of material is what are those changes in the DNA, how do they lead to changes in the proteins, and how do they happen? How is our DNA change? How is it mutated? What are some chemicals that cause this? So one thing to think about is the types of mutations that can occur. Somatic mutations versus germline mutations. So we've mentioned before, somatic cells are basically all of your cells except for the cells that will become your reproductive cells, so the eggs and the sperm, right? So if you have a mutation in a somatic cell, you cannot pass that on to your offspring, right? So if you get mutations in your skin cells or in your liver cells or your brain cells, those mutations will not be passed on. If you get a mutation in your germ cells, right, and the cells that are going to develop into the eggs and the sperm, that can be passed on to the offspring. Now, when it comes to looking at human disease, right, most of the mutations that we look at are mutations in somatic cells, cells that can't be passed down, things like cancer and these other things. Now, if you're doing research and you're trying to create mutations and you're trying to create a line of, let's say, mice that can be infected by a particular virus or whatnot, you want to create mutations in the germ cells, right? Because you want to create mutations that can be passed down to the offspring so that you have a whole bunch of mice with the similar phenotype. So that's one thing to think about, is where the mutation is occurring and whether that mutation can be inherited. Okay. Mutations cause changes, or can cause changes in protein function. We will talk about some examples of how a mutation or a change in the DNA can have no impact. It doesn't actually change the protein at all. But if it does change the protein function, there are two typical ways that this can happen. One is a loss of function, As the name suggests, this means that the protein no longer functions. This can be a complete loss of function. So the protein doesn't work at all, or is not even present in the cells anymore. It doesn't, there's no function for that protein whatsoever. Or it could be a partial absence of the protein function, right? So in sickle cell anemia, for example, the hemoglobin protein, it works a little. It just doesn't work as well. So it's a partial loss of that protein's function. So it's not completely missing, it's just not working very well. Loss of function mutations are recessive acting. So what does that mean? You have 2 copies of most every gene, right? 2 copies of all of your chromosomes, right? Unless you're a male, you only have 1 copy of the X. But for every other chromosome, you have 2 copies. So if one of the copies contains an allele with a loss of function mutation, you still have 1 good copy, right? So if you're heterozygous for the sickle cell allele, you still have 1 completely normal hemoglobin gene that can make normal hemoglobin protein, and so typically you're not going to have a phenotype there. So you're going to be fine. But if you lose both functioning copies of that particular gene, then you're going to see the mutant phenotype. The other type of change we can have is a gain of function. So these are a little bit more difficult to kind of think about, but this can definitely happen. So instead of the protein basically no longer functioning, this is when the protein gains a new function. And you may think, how can a protein gain a new function? Well, we talked about one example a few weeks ago about how we had a translocation that created this fusion protein that was like 2 proteins together that could do new stuff. But more commonly what happens is the protein is produced in a new place, or it's produced at a new time, or it's produced at a different level. So if a protein is important for regulating something, for example, let's say a protein is important for regulating growth. So there's a lot of different proteins in your cells that are important for regulating growth of different things, right? So if you notice, your arms are typically the same length and your ears are the same size, and so we have to make sure things are growing at the right times and they're not growing too fast or too slow or too big. So there's a lot of genes and proteins that are important for regulating growth. Well, what if we have a protein and its whole job is to regulate when these cells should grow and divide, and it waits to receive a signal that says, okay, it's time to grow. Well, what if there's a mutation that causes this protein to no longer need a signal? So now it just grows whenever it wants to. It tells the cell, grow now, even though it's not supposed to, right? So a gene that encodes a growth factor receptor might cause the mutated receptor to stimulate growth in the absence of the signal. So if there's a mutation in that gene that was a gain of function, the gene might be working when it shouldn't be, when it's not supposed to. Gain of function mutations are typically dominant acting, and this will come back a lot when we talk about the genetics of cancer. But think about, again, if you have a gene that's important for regulating the growth and you have 2 copies, and 1 copy is making a protein that's doing its job and regulating everything when it's supposed to, and the other one has a mutation that's causing it to just go haywire and tell the cells to grow and grow and grow, it doesn't matter that the original copy is still working properly, right? The mutated copy is in overdrive, so it's going to be dominant. Its phenotype is going to be showing up even though the normal copy is still present. Okay, so let's talk about some mutations at the DNA level, at the molecular level. One type of a mutation is called a point mutation. A point mutation is the substitution of one base for another. There are 2 types of point mutations, a transition and a transversion. So remember I told you that you would need to know what's a purine and what's a pyrimidine. That would come back. Well, here it is, right? A purine to a purine is a transition. A purine to a pyrimidine is a transversion. A pyrimidine to a pyrimidine is a transition. A pyrimidine to a purine is a transversion, right? So we have to remember which ones are purines and which ones are pyrimidines to figure out whether we have a transition or a transversion. So a transition is when a purine changes to a purine, so A to G or G to A, or a pyrimidine changes to a pyrimidine, so T to C or C to D. And a transversion is when a pyrimidine changes to a purine or a purine changes to a pyrimidine. Okay, so how can this affect the protein? right? So if we change a single base, there are a couple different things that can happen. One is called a missense mutation. So a missense mutation is when we change the amino acid. So that's what happens in sickle cell. We just change that one amino acid by changing a single base. So this may alter the protein function. So again, it alters that function of that hemoglobin pretty drastically. It may not alter the protein function at all. There may be two amino acids that are pretty similar. They have similarly charged side chains, and so when we switch them out, it doesn't really change the function of the protein much. So that could be a neutral missense mutation. A non-sense mutation is when we change a codon into a stop codon. So we change a codon from a codon that codes for an amino acid to a stop codon. So for example, if we have a GAG codon and the DNA changes such that now the RNA reads UAG, now instead of an amino acid there, the translation will stop. We could have a silent mutation. So remember, when you look at that genetic code, there's a lot of codons that actually code for the same amino acid. So you could have a mutation that changes the DNA, which changes the messenger RNA sequence, but actually the amino acid sequence stays the same. So if we have AGG in the coding strand and it's changed to CGG in the coding strand, those both code for arginine. So that actually would not change the amino acid sequence. Okay, so we have a missense, which is when we change the amino acid to a different amino acid. We have a non-sense, which is when we change the amino acid codon to a stop codon. We have a silent mutation, which changes to a different codon, but it's the same amino acid. And so then the last one would be if we change a stop codon to an amino acid. And so that's what we call read-through. So you just keep on reading right through the end of that amino acid sequence, right? So instead of stopping, we're going to keep going. So here's a figure that shows us basically the same information. So initially in our DNA, in the coding strand of the DNA, we have TCA, which means our messenger RNA has UCA, which codes for a serine amino acid. So if the C in that coding strand is changed to a T, now our messenger RNA is going to have a UUA, which is going to code for a leucine. So that would be a missense mutation, going from serine to leucine. If that C in the DNA changes to an A in the coding strand, that would mean our messenger RNA would then have a UAA, which codes for a stop. So that would be a change from a serine amino acid to a stop codon or a release factor, so that would be a non-sense. And then the last would be if we change the A at the end of the codon in the DNA coding strand to a G. Well, that goes from a UCA in the mRNA to a UCG, which it still codes for serine, so that would be a silent. So the three types of effects of the point mutation that we have here are missense, non-sense, and silent. And then there's also the read-through. Then I want to just tell you that it can happen that you have a mutation to something and you can have another mutation that reverts it back to the original, right? So you can have an exact reversion. We can mutate an AAA to a GAA, and then it could get mutated back to AAA. So that would be an exact reversion. We could have an equivalent reversion, which is also known as a suppressor mutation. So for example, if we have a UCC codon that has been changed to a UGC codon, and then it's changed again with a second mutation to an AGC codon, right? That is ends up back at a serine codon, but it's due to a second mutation. So it's not the exact same codon we started with, but it still codes for a serine amino acid. There are two types of suppressor mutations. You can have an intragenic or an intergenic. So intragenic is within the same gene. Intergenic occurs in a different gene. So here's an example of the intragenic suppressor. right? So we ended up with, we have TTA in our coding strand of the DNA, which gives us UUA in the mRNA, which codes for leucine. Then we have a mutation, which gives us TTT in the coding strand of the DNA, which gives us UUU in the mRNA, which codes for phenylalanine. Then we have a second mutation, which gives us CTT in the coding strand of the DNA, which gives us CUU in the mRNA, which codes again for leucine. So we've had two mutations, but we end up where we started. Okay? Now, an intergenic suppressor would be a mutation in a different gene. And these actually intergenic suppressor mutations are actually used a lot in research to help us understand the relationship between proteins and things in different phenotypes. So for example, if we have a wild type or normal fly that has red eyes, and we mutate this fly so now it has white eyes. So now this fly has a mutation in a gene that's causing its eyes to be white. Well, if we cause additional mutations in this fly, it's possible that we could mutate in a second gene, and by mutating the second gene, we could revert the phenotype back to normal. So then he would have red eyes, right? And so then we could try to figure out what gene did we mutate, right? Maybe we mutated two genes that are binding partners, and when we mutated the first gene, they couldn't bind together, but when we mutated the second gene, well, now we've altered its configuration so they can bind together again. Maybe there are two genes involved in the same pathway, and they have some sort of epistatic relationship, such that when we got rid of one, we had one phenotype, but now that we got rid of both, we could get back to that original phenotype. Right? So we can have mutations in second sites or in different genes, and by trying to understand the relationship between the two genes, that can tell us a little bit about the mechanism of this particular phenotype. Another example of a reverse mutation would be in a tRNA. So let's say our initial mutation was a nonsense, so we've mutated a DNA to codon into a stop codon, so that when translation begins, we're going to stop prematurely because we have this premature stop codon. Well, what if there's a second mutation, and this second mutation actually mutates one of your DNAs that encode a tRNA? Now you actually have multiple copies of these tRNA molecules, so if we change one, if you mutated one of them, it wouldn't affect every one because we have backup copies. But let's say we mutated one of these DNA molecules that encode this tRNA. And now this tRNA, the anticodon is, instead of being the proper anticodon, it's an anticodon that can recognize a stop codon. So now, instead of stopping at this premature stop, we're going to keep going. We're going to insert tyrosine. Right? But you have to remember that if this happened, it wouldn't just affect this one protein that was having this nonsense mutation. It would also insert tyrosines in other proteins where there should be a stop codon, right? So it could have additional phenotypes or additional effects. One thing I want you to realize is that your cells experience tens of thousands of mutations per cell per day. All the things that we encounter in the environment many of them can cause mutations. Just the normal chemical structures of bases and DNA replication can cause mutations. Mutations are always happening. I'm going to tell you a little bit about the mechanisms of mutation and some different chemicals and some different things that can cause mutations. And I don't want you to spend a lot of time trying to memorize these or trying to remember all the details of the chemicals or the chemical reactions that cause mutations, but really think about what would happen if this mutation occurred and then the cell underwent replication. Because we're going to talk about DNA repair next, and if we have a mutation and then the cell repairs it, well, that's not really a problem. But if we have a mutation and then the DNA is replicated before it can be repaired, well, that can be a problem, because then once we've replicated the DNA, now we could be on the road to making this mutation permanent, and then the cell wouldn't even know that this was the incorrect base, right? All right, so let's think about some of these types of mutations that occur. Spontaneous point mutations. These are mutations that happen spontaneously. There's nothing that we really do to cause them. There's nothing that we really do to prevent them. They're just based on the chemical stability and structure of DNA. One is a depurination. Depurination removes the glycosidic bond at either the G or the A bases. So we're getting rid of a purine. Depurination, basically just a purine is removed. So you still have the backbone, but you don't have the A or the G. Deamination. Deamination of cytosine results in uracil. So if you remove an amine from cytosine, it causes a uracil, or it creates a uracil. And then wobble. Remember we talked about wobble as it relates to tRNA, mRNA binding and the codon-anticodon pairing, but sometimes wobble can happen in DNA replication. And if the bases are pairing wobbly during DNA replication, then that could insert the wrong base. All right, so let's walk through these in a little more detail. So depurination, that's the removal of the bond between the base and the sugar. So in this example here, right, we've had a depurination event and we removed that A. In a mammalian cell, in about 20 hours, there's about 10,000 depurination events that occur. So you may think, well, what's the problem, right? If here's a double-stranded molecule, what if we remove that A, right? Well, as long as you can catch it, well, let's just put it back, right? Because you look and you say, well, there's a T across from it, so an A has to go here. And yes, your cell can repair that, and I'll tell you about that in the next part about DNA repair. But what happens if we go to replicate this DNA before we fix it? Remember, in DNA replication, we're going to separate the two strands. And when we separate the two strands, now we're going to have a strand basically with a hole. And the DNA polymerase is not going to know what to do. So here is an image showing that. So here we have our depurination event. And in this example, we removed a G. So we have our apurinic site. So now the first round of replication is going to begin. Okay? So over here in our top strand that has our depurinic site, then we are going to have an empty spot when we go to replicate it. And oftentimes when the DNA polymerase is replicating DNA and it doesn't know what to put, it's just going to put an A. That's just like DNA polymerase's favorite base. So it's like, oh, I don't know what goes here, let's put an A. Right? So it's going to put an A across from that empty spot. But really across from that empty spot was supposed to be a C. But on this strand here, there was no change. So when this is replicated, it's completely normal. So now we have two daughter cells. In one daughter cell is normal, and in one daughter cell we have an A across from an empty spot. So now let's undergo a second round of replication in that abnormal daughter cell. So in that abnormal cell, right, let's assume we have not repaired this issue. So we've separated our strands, and now across from this A, the DNA polymerase is going to put a T. So after 2 rounds of replication, what used to be a GC base pair is now a TA base pair. Right? Because that apurinic site was not fixed, was not repaired before the initial replication began. Right? So this is what I mean by focus more on the effect of the mutation. If we removed a purine and we replicated the cells before we were able to fix it, how would that change the DNA sequence? So here's the DNA deamination of cytosine. So deamination of cytosine creates a uracil. So again, if we don't fix that, right, normally across from a C in replication we would put a G. But if the C has been changed to a U, across from a U, we are going to put an A. So instead of the CG base pair, we're going to have a UA base pair, which after 2 rounds of replication will become a TA base pair. So again, after 2 rounds of replication, you would have a completely different base pair. And I would recommend that you pause and try to write that out for yourself and see if you can see how if we changed a C to a U, how does that make us change from a CG base pair after 2 rounds of replication to a TA base pair. Wobble base pairing also can cause replication errors. So if the DNA polymerase, instead of putting a G across from a C, it puts an A. That's going to cause a problem. If instead of a across from a G, A across from a T, it puts a G, that causes a problem. So again, after 2 rounds of replication, we're going to have a problem. So let's look at that wobble base pairing, right? So here we have TTCG and AACG, TTCG and AAGC. We're going to separate those 2 strands. On the bottom half, everything is replicated perfectly fine. But on the top half, right, we see that across from that T, the DNA polymerase accidentally put a G. So we have a G across from a T in that top section, right? And so now when we separate those 2 strands, right, if the cell doesn't realize that that's an incorrect pairing and we separate those 2 strands, well, the TTCG, that might be replicated properly. But the AGGC, now when the DNA polymerase replicates that across from that strand, it's going to put TCCG, which is not what it's supposed to have. It's supposed to be TTCG, not TC. So we've changed a TA base pair to a CG base pair. Right, so for these spontaneous changes, I would definitely recommend spending some time walking through how does that lead to a change in your DNA sequence after 2 rounds of replication, right? We have the incorrect pairing, we separate those strands, and then how does that change after 1 round and after 2 rounds of replication? So those are all spontaneous. They happen naturally based on the chemical structures of DNA and DNA polymerase's abilities, right? Then there's also mutations that are induced by chemicals. So these are just some examples of chemically induced, or things that can induce mutations: base analogs, alkylating agents, deamination, hydroxylamine, oxidative reactions, and intercalating agents. So let's talk through a few of these. Base analogs. Okay, so we have a 5-bromouracil. So 5-bromouracil is a base analog, right? That's not a normal base. This 5-bromouracil normally pairs with adenine, but it's more likely than a normal thymine to mispair. It can also pair with guanine. 2-aminopurine normally pairs with thymine, right? But it can also pair with cytosine. So if we have a 5-bromouracil that's incorporated into our DNA, then in replication, sometimes instead of putting an A across from that 5-bromouracil, we are going to put a G, or the DNA polymerase will put a G. Okay? And so then, if we put a G across from that 5-bromouracil, and then we replicate it a second time, that G, across from that G, we're going to put a C. So we'll go from having our normal base pair to having a mutation, a mispairing, because of these base analogs are more likely to mispair, which will lead to changes after 2 rounds of replication. So here's an example of this, right? So let's say 5-bromouracil is in the cell. And so when the DNA is being replicated, the DNA polymerase incorporates that bromouracil, right, and across from the A instead of a thymine. Okay? So we've incorporated the error. Then now we're going to replicate it. So now when we replicate that DNA, we separate those 2 strands, and that bromouracil pairs with a G instead of the A that it's supposed to pair with, right? So after 1 round of replication, now we have a mispairing, right? We've mispaired that bromouracil with the G. Now after a second round of replication, that G will be properly paired with the C. But that's not what's supposed to go there, right? What's supposed to be there is an AT base pair. But now instead of an AT base pair, we have a GC base pair. So because these base analogs are more likely to have mispairings than other normal bases, you're more likely to have these sorts of mutations. Here are some other examples. We have alkylating agents. So alkylating agents, for example, EMS is an alkylating agent. It changes guanine to ethylguanine. Ethylguanine will pair with thymine, which is obviously not what G's are supposed to pair with. Like G's normally pairs with C's, but ethylguanines pair with T's. Deamination, so like nitrous acid, can deaminate cytosine to make it a uracil. So again, normally cytosine pairs with guanine, but uracil would pair with adenine. So if we change our cytosine to a uracil, right, we're going to change our base pair after replication. And hydroxylamine changes a cytosine to a hydroxylaminocytosine, right? And cytosine normally pairs with guanine, but hydroxylaminocytosine pairs with adenine. So now we can create a CG to a TA change. Oxidating agents, so oxidating agents, the oxidative reaction converts guanine into 8-oxyguanine. 8-oxyguanine pairs with adenine instead of cytosine during replication. So this adenine can now pair with thymine during the next round of replication. So that takes 2 rounds of replication where we have this oxyguanine pairing with adenine, then we replicate it again and now adenine's pairing with thymine. So that's going to create a GC to TA transversion. So those are just some examples of the chemicals that can cause changes. So again, I don't want you to memorize that oxyguanine or oxidative reactions create oxyguanine and oxyguanine pairs with adenine. I want you to be able to say, if I told you that, oxyguanine is a guanine that's modified and now it can pair with adenine, what would happen in DNA replication? What would happen after 1 round of DNA replication if this mispairing occurs? What would happen after 2 rounds of DNA replication, right? So those are point mutations. The other thing I want to tell you about are frame shift mutations. So chemicals like proflavin, ethidium bromide, acridine orange, they are large planar molecules that slip in between the base pairs of DNA. So DNA, are kind of like, the base pairs are kind of like rungs on the ladder. But if we slip these intercalating agents in between, well, now the base pairs are a little bit too far apart. And so when the DNA polymerase goes to replicate this DNA that have these intercalating agents in here, it's not really sure what to do. It's like, well, these 2 bases are really far apart. Is there supposed to be something in between here? Is there supposed to be an extra base? Maybe I should add a base, right? Because these 2 bases seem like there's something that should be in between these 2, right? So here's an example of what might happen if we had a frame shift mutation. So a frame shift mutation is the insertion or deletion of 1 or more base pairs in the gene. And it's called a frame shift because this will shift the reading frame. So if just, for example, we have a protein that has a string of alanines, right? So here we have we have 7 different codons that all code for alanine. And there was a frame shift mutation that inserted an A. So if we inserted an A, well, now instead of a whole bunch of alanines, we have a few alanines followed by a serine and then a bunch of cysteines. And anything after this will also be changed. This could also happen if we deleted. So in this bottom example, if we deleted a G, right, instead of a whole bunch of alanines, now we have a bunch of alanines followed by leucines. Now you can also have suppressor mutations in frame shift. So if we add a base and then delete a base, that'll get us back to our normal reading frame. So in that example, we have a couple alanines and then we have a few changed amino acids. But once the deletion occurs, we're back to that normal reading frame. Okay. Now you can have frame shift mutations that insert 2 bases or 4 bases or 6 or 5 bases or more. But if you insert 3 bases, you're gonna alter a few of the amino acids in the middle there, but you're not going to change the entire reading frame, right? If you insert 3 bases, while you're going to change some of the codons, the reading frame in general will stay the same. Or if you delete 3 bases, it's like deleting an entire codon, right? Other ways that frame shift mutations that can be caused, in addition to by chemicals, is template slippage. So as your DNA polymerase is replicating the DNA, right, sometimes it can, slip. Sometimes, especially if there's a long string of repeats, a long string of a repeated region, it can have a situation where it doesn't know whether or not it's already done those A's or done those T's. So here's two examples. One example is where the newly synthesized strand loops out, and we're actually going to add an extra A. So we're going to replicate that T one more time than we should. So we have an extra A on the new strand. We could also have a situation where the template strand loops out. So one of those T's kind of gets hidden and is not replicated into an A. And so now we have one less base on our new strand. We could also get frame shift mutations by unequal crossing over. So remember we talked about DNA recombination a lot in Unit 2. And we talked about how unequal crossing over can cause insertions and deletions when we talked about insertions and deletions of the chromosome. But if they are almost exactly right, but not quite, that can still cause a problem, right? So if those homologs that are lined up and they're not completely exactly lined up properly, we could get a frame shift mutation. We could get the insertion or deletion of a few bases, which actually could cause a problem if it's in the middle of an open reading frame. So here we have an example of a repeat region causing a frame shift mutation. So in this example here, you see the two original DNA strands in gray, right? They are separated, and now we're just showing one of the original strands. The newly replicated strand is indicated in red, and you can see that in this newly replicated strand, there's a hairpin that forms. Remember, so hairpins is when you have these regions that have the inverse repeats and they're able to bind to themselves. And so now we have the formation of a hairpin in the new strand, which means when this hairpin forms, the DNA polymerase is going to replicate this same region twice. So this region of the DNA is replicated more than once, which is going to make this region longer. So this leads to more repeats in the daughter cells. And actually this sometimes leads to particular diseases or disorders that we know about. One example of that is Fragile X syndrome. These individuals have a trinucleotide repeat CGG on their X chromosome. It's more frequent in males than in females, and symptoms of Fragile X syndrome range from learning disabilities to severe disabilities, as well as behavior, attention problems, and autistic behaviors. They also have physical symptoms including loose joints and a very long jawbone. And this is due to a trinucleotide repeat that can expand and get longer due to these template slippage and these repeat regions, right, in the DNA. So here are some other examples of diseases caused by these nuclear repeats that can be expanded in DNA replication. We mentioned Fragile X syndrome. We've also talked a little bit about Huntington's disease. So we've talked about point mutations. We've talked about frame shift mutations. The other big type of mutation that can occur is chromosome breakage. So chromosome breakage is when your chromosomes break, when we have double-strand breaks in your DNA. Chromosome breakage leads to aberrations by breaking the phosphodiester bonds. X-rays can do this. They can also damage bases and cause point mutations. X-rays cause the most damage in dividing cells. So they are going to, the chromosomes are more condensed in these dividing cells. Remember in prophase, the chromosomes condense. And so therefore they are more prone to breaking due to these X-rays. So this is initially, and this is still used somewhat in cancer treatments, because if we're going to shine X-rays at a tumor, then it's much more likely to attack the cells that are actively dividing versus the surrounding normal tissue that's not dividing as much. UV light, we all know UV light causes mutations. So what kind of mutations does UV light cause? It causes something called a thymine dimer. These are pyrimidine dimers, particularly thymine dimers are the most common. These dimers distort the helix and they inhibit replication. So basically the UV light causes covalent bonds to form between two adjacent thymines. And this is a problem because if we have these thymine dimers, it's going to distort the double helix, and the DNA polymerase is like, I don't know what to do, I can't replicate that. So cells with thymine dimers the DNA can't be replicated, which means that cell can't divide. So we either need to fix the thymine dimer or we need to find a backup polymerase that can replicate that DNA even with a thymine dimer, right? And that can cause additional problems to use kind of the backup polymerases. I'll tell you about more that. There's more in a minute. But xeroderma pigmentosum is an example of a human disorder where they cannot repair these thymine dimers. So when they get thymine dimers as a result of UV light, they can't repair them. So their cells have to decide what to do. Should they continue on and try to replicate this DNA even though it has all these thymine dimers, or should they undergo cell death, basically? And if they choose to continue on and divide, they can't use the normal DNA polymerases. They have to use the backup DNA polymerases, which are not as good and are much more likely to cause errors. Okay, so let's do a couple of samples to look at some mutations. So here is a normal DNA sequence and a mutant DNA sequence. So the first thing that we want to figure out is what type of mutation is this? So I've written this out on my paper here. And if we look at our DNA sequence, let's see, let's try to figure out where the problem is. So this looks the same here. Oh, I think I found it. But it's still the same moving here, so it's just a single base that has changed. So this would be a point mutation, right? So then the next question would be, is this a transition or a transversion? So if we're going a C to a G, right? A transition is when we go purine to purine or pyrimidine to a pyrimidine. A transversion is when we go purine to pyrimidine or pyrimidine to purine. So this will be a transversion. Okay. Then the next question would be, what does this do to the amino acid sequence? Is this a missense, a nonsense, a silent, or a read-through? So in order to figure that out, we have to figure out what is the reading frame of this particular piece of DNA, right? And these are both the coding strand, right? So we're looking at the coding strand of the DNA. So if we want to write out our mRNA, right, it would be, so we're going to do the normal, CAA, UGC, CAG. Okay, so here's our mRNA, right? Because remember, if this is the coding strand, the messenger RNA is going to have the same sequence as the coding strand, just switching out our T's for U's. Okay, so we want to find the reading frame. So for the reading frame, we're going to look 5 to 3 and find our first AUG. So it looks like here's one here. So AUG, CCA, GAU, UAC. Okay, so then where was our mutations? Let's see, it was this here, right? So that UAC was changed to a UAG. So let's see if we can figure out what happened here. So UAC is tyrosine, UAG is a stop. So it looks like that this was a change from an amino acid to a stop codon, which means this would be a nonsense mutation. So it's a change in a single base, so this is a point mutation. Is it a transition or a transversion? Well, we're going from a C to a G, so that would be a transversion. This is a change from a CG base pair to a GC base pair. And then here we have in the protein, right, this would be a nonsense mutation because we changed from a UAC codon to a UAG codon. Okay, here's one that's a little more fun. So in this one, we have two proteins, and we're going to try to figure out what has changed in the DNA sequence to make a changed RNA sequence to make these changed proteins. All right, so I'm going to write out my normal protein. We have a methionine, a trip, cis, and then it looks like our mutant protein is longer than our normal protein, so I think after this cis there is a stop. Right, so then in our mutant protein we have methionine, trip, phenylalanine, and glutamate. Okay. So the next step is we need to write out the codons that can be used to create these amino acids. So methionine is AUG, trip is right here, UGG, cis right here, UGU. And now there's more than one that can make cysteine, so I just write the other possibility below. And then our stops, there's UAA, UAG, and UGA. All right, so let's go down here to, all right, again, methionine, AUG, trip is UGG, phenylalanine is over here, UUU, and then UUC, and then glutamate is down here, GAA and GAG. Okay, so what changed from looking at one to the next? Well, AUG and AUG, that's the same. Then UGG and UGG, that seems to be the same. So then now let's try to figure out what happened. So this U and that U is the same, but then this is different. We have a G here, we have a U here. So the question is, did we insert a U or did we remove the G? If we inserted a U, that would make us have U, U, G, and then a U or a C. So if you look at this UUG, that doesn't match what we have down here. There's no way that you could have a phenylalanine with a UUG, right? So again, if we inserted a U, that would give us UUG, right? UUG. And then if you look down here at your phenylalanine, that doesn't work. So it can't be an insertion. It looks like we deleted this G. So if we deleted that G, then over here, this U would be that U. And over here, the stop starts with a U, so that has to be one of these. Then the G, and then the A. So we can rewrite our normal mRNA and our mutant mRNA, right, now that we know that this was a deletion. Okay, so here is our normal would be AUG, UGG, UGU, UGA. And then our mutant is AUG, UGG, UUU, GA. And we don't actually know what's here because we don't have any more information. So we know that this is a G and this is an A, but this could either be an A or a G, right? So if you want to kind of confirm that this is actually what happened, let's write out our normal DNA and see if we can, so AUG, normal mRNA, UGG, U, then delete our G, U, UGA, and that matches up here. AUG, UGG, UUU, GA blank. So again, it was a deletion of this G. So this is a frame shift mutation. So here is our potential amino acids, potential codons for these amino acids in both the normal and the mutant. And so then we're going to line them up. So the AUG did not change, the UGG did not change, the next U is the same, but now we have a G in the normal protein, but a U in the mutant protein. So the question is, did we insert a U to get from the normal to the mutant? And if we had inserted a U, then our mutant would be UUG, but we don't see that in the mutant. So it has to be that we deleted the G, and now that we get UUU, and then GA, and we don't know what comes after that. Okay? So that is our discussion of mutations.